like to welcome you all here this evening. It's great to see so many people from the community and students and faculty tonight. Welcome everybody to the college. My name is Hai, and I'm a biology instructor here at the college. And it's my pleasure to be here tonight to introduce Dr. Leonard Foster, who's come to speak to us as part of a partnership with Genome BC. And the College of the Rockies has worked with Genome BC for the past 5 years, bringing speakers to the community. Enjoy, and it's something that we look forward to hosting every year. It's an excellent opportunity for our students and for our community to get to hear about some current research and really exciting topics. So thank you for coming. Dr. Foster grew up in McBride in, well, sort of northern British Columbia, oldest son of 2 teachers, and was involved in science fairs throughout his youth, and ended up making contact with Drs. Winston and Slesser at Simon Fraser University. And he joined them working in their laboratories while he did his undergraduate degree at Simon Fraser University, and then moved on to the University of Toronto, where he completed his PhD. After that, he moved to Denmark and spent a few years there learning about a new emerging field of study called proteomics. And then in 2005, came back to the University of British Columbia and started his current position where of associate professor in the Department of Biochemistry and Molecular Biology. He's also the director of the Center for High Throughput Biology there. Dr. Foster's research focuses on how bacterial and biopathogens manipulate the cells of their hosts, that include honeybees and humans. And he's also been a part of a large effort in Western Canada to try and develop bees that are better able to resist diseases. And that's what he's here to talk to us about this evening. There will be an opportunity for questions at the end of the presentation, so please save those questions and comments for that time. And please welcome Dr. Leonard Foster.
Okay, thanks very much for the introduction, Andrina. Can you hear me at the back? Okay, good. All right, I wanted to talk about a few things as background information first. So first of all, the organization that is sponsoring me to come here is Genome British Columbia. They're a provincial funding agency that funds genomics research, which is anything to do with genome or other areas of the complement of genes that we have in our bodies. And they don't just fund human research, they fund research in agriculture and forestry and many different areas. And they also have quite a mandate for outreach. So what I hope to do today is to tell you a little bit about my research, and Andrina mentioned a little bit about the big project we've been involved in. And I'll do that first and try to keep most of the technology out of it, but I'll explain a little bit about the techniques that we use and why they are allowing us to do some of the things for bees that were not previously possible. And then I'll get into a couple of the other areas of bee biology that I actually don't do any research on myself, but are quite topical, and I'm sure a lot of you have been hearing about them, and including ending off with a bit of a talk about the neonicotinoid class of insecticides, which seems to be all you ever hear about now if you read about bees in the news. Anyway, I want to tell you a little bit about the work we've been doing because we feel it is probably the way that bee farming, apiculture, is going to have to go in the future in order to stay ahead of the diseases that continually plague bees. So, bees, I had a question first. How many of you are beekeepers? Okay, a few, good. So most people probably appreciate this quite well now, but bees do many things for humans. They obviously produce honey, but from an economics point of view, they have far greater value to us in their pollination activities. So BC, the crops that are most relevant to BC are shown here. So we have cherry, apple, raspberry, blueberry, and cranberry, low bush cranberry. And these together have a value for BC somewhere around $300 or $400 million. And all told, honeybees are responsible for about 90% of the pollination in these crops. These are, BC is actually, per capita, is actually one of the major users of bees for pollination activities. Most of the rest of the provinces, the bee activities are confined to honey production, but Ontario has a lot of pollination, and the granddaddy of pollination activities in Canada is in the production of hybrid canola seed in southern Alberta. So about 80% of all hybrid canola seed produced for the world is produced in southern Alberta, and honeybees play a critical role in the pollination of those crops. So this is the production of the seed that's then planted to make the seed that is the oil is extracted from. So it's one step away from the oil production, canola oil production. But the reason that we're hearing about bees so much lately is really shown in this graph. So these are stats for Canada and the US, but very similar trends are seen for the rest of the world, where developed apiculture is anyway. So prior to 2005, the average beekeeper would expect to lose about 12 to 15% of their colonies every winter. And there's lots of reasons for those losses, but this was sort of the long-term average leading up to about 2005. Then in the winter of 2006, 2007, there's stories starting in the southeastern states, but really exploded across all of the continent and the rest of the world, where beekeepers were reporting losses that were up towards 30% or so. And about that time, or not just about that time, but as a reason to explain those losses, various groups came up with this thing called the colony collapse disorder. And I'll have a little bit more to say about that, but for the most part, colony collapse disorder doesn't really exist. The causes for these losses are mostly known and reasonably well defined, and I'll explain them later on. But the point of this slide is really that those losses have remained more or less at the 30% level for several years. Although if you plot a trend line here, you do see that it's decreasing slightly, and that may be encouraging, but you can see there's also a lot of noise, so it goes up and down. So we're not out of the woods yet. And if we were out of the woods, it would be through no fault of our own. The bees basically turned around themselves. But with this colony collapse disorder, there was it was very ill-defined, and it was an easy way for people to find a reason for their losses, the bees that were being lost, but it wasn't really based on a lot of hard data. So over the last, this is data from about 2011, 2012, but this is a reflection of what beekeepers thought they were losing bees to over the winter of 2010, 2011, and 2011, 2012. And you can see the vast majority of them were being lost to mites, and I'll have lots more to say about mites. There are other diseases in here. Nosema is a fungal disease. American foul brood is a bacterial disease. There is also this term weak queens, which usually actually means that the colony succumbed to a disease, but it was probably because the queen was not all that well adapted, or her genetics didn't allow the bees to survive whatever the disease was. A lot of losses are due to just harsh winter. Nutrition means, unfortunately, that the beekeeper didn't stay on top of their bees and make sure they were fed enough. And then there's a few other reasons. And pesticides is not insignificant here. But again, these are losses that the beekeepers attributed to to the causes. I would say that this is a survey, and with all the caveats that go with survey data, we can't really make many hard conclusions about this, except I think it's pretty safe to say that diseases are by far the major cause. And the key thing is that they're known diseases. So these are, if we had done this survey probably 4 years before, a lot of these losses would have still been attributed to this colony collapse disorder. But as everybody started losing bees, they started looking at bees a little bit more carefully and trying to monitor what the what the cause of the losses was or were. And really what emerged was that the known diseases or pests of bees seem to be causing most of the losses. Okay, so that's what some of the what was causing some of the losses. I mentioned the mites. So there's a few mites that infect bees or attack bees. By far, and it's hard to underestimate or understate just how much more of a problem this mite in particular is compared to everything else. But this mite is called Varroa destructor. It's a parasitic mite that basically sits on the back of the bee and feeds on the bee's hemolymph for the bee's blood. And that obviously weakens the bee. But actually what the mite does as well is it acts as a vector for several viruses. And this is much the same way that mosquitoes are a vector for malaria. The mite, when it bites or starts feeding on the bee, transfers several different viruses to the bees. And the viruses, I would claim, are probably at least as damaging to the bees as the mite feeding on the bee is. That being said, if you deal with the mites, for the most part, you will have dealt with the viruses. So the mites are really still the key here. There's also bacterial diseases, and they've been around with bees for a long time, and there's antibiotics that we can use to treat these, but as with many or all areas of agriculture, even human medicine, the more we can get away from those kind of chemicals, the better, while still remaining effective. And then there's a fungal disease called a microsporidium, or something called, or more specifically, nosema. Bees have many different ways of fighting these types of pests and pathogens. They have what's called an innate immune system, which is very similar to one of the immune systems that humans have. But I'm not really going to talk much about that today, because one of the really fascinating things about bees, and one of the reasons that bees have been studied for so long, is their social behaviors. And in particular, with respect to fighting off diseases, they have something called social immunity. So these are behaviors that allow them to resist disease, or in some case, eradicate disease. So if you think about it, this is sort of analogous to humans building hospitals. And forget about what's actually done in the hospitals, but we do things on a social scale there that allow us to fight off the diseases. So one particular, I'll mention one more thing. So one particular behavior that bees exhibit that is particularly relevant for resisting disease is something called hygienic behavior. And this is a reasonably well-defined behavior, but it's very hard to test for, and I'll have much more to say about that in a minute. But all of this is to say that bees have some of these defenses. They don't always work, but there's a lot of variation in bees. So some bees are able to resist these diseases very well, and they do it through a combination of different things, but including hygienic behavior and other social immune characteristics. And other bees are not very good at resisting disease because they don't have these. But what we're not able to do very effectively is select for those bees that do have these special behaviors. There are tests for it, and they can be used, but they're just not used by the industry very commonly because they're very hard to do. And so our goal is to try and come up with different ways of selecting bees that have these special behaviors that allow them to resist disease. Should state up front that we're not trying to genetically modify bees here. We're just looking for a way to look at the natural variation in bees and select from that the bees that are going to be most disease resistant. So, again, to jump a little bit, I won't have too much to say about the technology, but it's worth understanding what I'm what technology we're using in order to understand how we're going about doing this selection for hygienic behavior. So I'll remind everyone, and I know there's a lot of first-year biology students here right now, so you're probably immersed in this right now, but every living thing has genes, and every living thing has a genome. Just to define what these different things are, a gene can be defined in a few different ways, and I think everybody probably has a concept of a gene. If you think about characteristics that we have as humans, things like eye color are defined by a particular variant of a gene that you have. But a gene can be defined as a heritable unit. So you inherit some gene from your parents, and you at the same time you're conferred with whatever the trait is that gene enables. But genes are also defined at the level of molecules. So a gene is also a sequence of DNA, and most of you probably know that DNA is composed of a set of 4 different chemicals all linked together, and they're referred to by this single letter set of A, C, G, and T. So these are the different heritable units that define us as an organism, and together all of those genes are known as the genome. So all of the DNA or all of the genes in our cells are collectively called the genome. In honeybees, there's about 253 million megabases, so 253 million of these different single-letter bases that comprise the honeybee genome. For reference, humans are somewhere around 3 billion. So bees have about 10 times less DNA in their cells than we do. And in that 253 million bases, there's somewhere around 14,000 to 15,000 different genes. But genes just hold information. The genes don't actually do anything all by themselves. And that's where the technology that I'm going to explain a bit more about, and Andrina mentioned. So genes really just hold information, and they allow the cells to make proteins. And it's the proteins that, for the most part, do all the work in the cells. It's proteins that give your eyes a blue color or a green color. It's proteins that allow you to move your muscles around. It's proteins that allow you to use to take honey and extract energy from it. So of those 15,000 genes in bees, they make somewhere greater than 100,000 proteins. And we don't really know how many, but that's not really important. I mentioned this, genes themselves aren't the things that are doing the work. But this now is the instrument that we use to measure proteins, and I won't explain very much about it. This is almost life-size, actually, on this screen. So the instrument comes up to about my chest. And what it really allows us to do is to measure the masses of things very, very accurately. So we can take a molecule and measure it to this level of accuracy to this level of detail with about 1 part in 10,000 accuracy. And then we can break those molecules up and identify parts of them. And again, that's as technical as I'm going to get for the lecture, but this is what the data looks like. So what we've done over the last now 7 years, I guess, is to collect bees mostly from across Canada, across Western Canada, but really focused on the West Kootenays, actually, and the Lower Mainland of BC, and measure their level of hygiene. So we have an assay, and I'll explain it, to test how hygienic bees are. And then we take those bees, we cut their antenna off, and we take those antennae back to the lab and inject them into the mass spectrometer that I just showed you. And this is what the data looks like. So we get these very, very complex patterns. And we look very hard at the data from hygienic bees. Where are we here? So data from hygienic bees and data from non-hygienic bees. And if you look very closely, you hopefully will find some difference in the hygienic bees versus the non-hygienic bees. And it's those differences, and it's just overemphasized with the little broom here, but we're looking for differences in the pattern between this hygienic bee and this non-hygienic bee. And when we find something like that, it gives us a tool, a handle on what the difference is between hygienic and non-hygienic at the molecular level. So this is very much analogous to you going into a hospital and having a blood sample taken. If you're pregnant, there's a whole bunch of tests that they'll do, including looking for trisomy, for example. And in all of those kinds of tests, this is the kind of thing that they're doing, except they're no longer looking at the complex data. They've focused down on the single molecule that they know to be different between trisomy versus normal chromosome 21 levels, or in this case, versus hygienic, in hygienic versus non-hygienic. So we're just looking for differences in these patterns. So again, I'll just skip this actually. I just wanted to highlight one last time that what we're doing ultimately here is looking for differences in the natural variation in bees. And the hope is that in the end, we'll be able to use these tools to select for bees that are naturally resistant to disease. And I'll show you how we're going to do that. So just a quick review of what selective breeding is. And this, I'm showing you bees here, but it really applies to any animal or plant. But you've got two parents. In the case of a bee, you'd have a drone versus a queen. And you breed them, and you generate a whole bunch of offspring. And you measure something about those offspring. So in this case, I'm showing you that we're measuring the yield of honey, because that's an easy thing to depict. But you measure the honey that each of these different families of bees makes at the end of a summer, and you observe under equal conditions that these bees made more honey than all the rest, and this bee here made second most. And so if you're interested in breeding bees that are efficient at making honey, then you might breed these strains of bees together and go through the process again, make more offspring from these bees and make the measurements again. But this process is, for one thing, very inefficient in bees. It takes a long time to do the test, but it's also really subjected to other things that are going on in the environment. So if there was, so as I said, it takes a long time to do the test, but if there's weird plants in the environment of this colony, then maybe that's the reason these bees didn't produce much honey, and they're too busy eating it. So what we're aiming to do is to bring this new technology that I described a little bit about to bear on this kind of a problem. And our approach to doing this for bees is new in the bee world, but it's something that's done pretty commonly in some of the other major livestock groups. For instance, dairy cows, this kind of a molecular approach to bees is the only way to do things now, or else you just don't stay competitive if you're trying to make milk. It's not done in bees yet, and so that's where we're going with this. So the process has been, or is depicted here. We started off working with normal selective breeding programs that were going through hygienic behavior testing. And so from that, we know which bees are hygienic and which bees are not hygienic. And then, as I said, we took those bees that were hygienic and the ones that were not hygienic as a control to look for signatures, these molecular signatures that corresponded to things that were resistant. And in the end, if we are able to use these effectively, we should be able to speed up the whole process so that we can do a selection in about 5 weeks. And eventually, this should allow beekeepers to select stock that is more resistant. And when I get to the end of my data, I'll show you that we think that's going to work. So this is how hygienic behavior is tested. So this, if you're not familiar with bees, a frame that would normally sit in a beehive. This brownish area here is bee pupa that have been metamorphed and are now in their cocoon and developing, and eventually they'll emerge as an adult. What we do, though, rather cruelly, is basically screw a bottomless tin can into that and then pour liquid nitrogen into the tin can. So you can see the condensation on the outside of the can here. And the liquid nitrogen kills the bees, or the developing bees that are in there. Then you take that tin can away and put them back into the colony. And in a colony that's hygienic, they will recognize that all those bees that were in there are dead from the freezing, and they'll clean them out, and they'll do that within about a 24-hour period, versus a non-hygienic colony basically will ignore it. They will not even recognize at all that there's dead bees in there. And why this is relevant for disease resistance is if a developing bee dies from a disease, it often is going to die at the pupal stage inside one of these capped cells. And for bees to be able to recognize that there's a diseased or dead bee in there and get rid of it, it allows them to basically keep a cleaner colony. They can take that disease out. They will literally pull that dead bee out and remove the carcass from the colony. And that process, if they do it over and over again and there's a disease present, allows them to get rid of the disease and keep most of the disease outside of the colony, thereby allowing the colony overall to resist that disease. So this is it's by far not the only trait that's of interest, but it's quite a well-studied trait and a trait that's recognized to be of value for beekeeping. If you have bees that are very hygienic, you can get away with not having to treat with antibiotics or some of the other chemicals to control disease. So, like I said, we were working with beekeepers who were breeding for hygienic behavior using this conventional test that I just described, looking at a lot of antennae from a lot of bees and looking for differences in some of them that hopefully would correspond with the levels of disease resistance or hygienic behavior in those bees. So the data that we ended up getting looked something like this. These are two particular protein molecules where the level of the protein on the bottom here corresponds or correlates quite well with the level of a hygienic behavior on the y-axis. So this is the kind of thing we're looking for. This protein would be considered a very good indicator of whether bees are going to be disease resistant or not. So if we then take this information or know this and look at a bunch of bees that we don't know if they're going to be disease resistant or not, for those bees that we find a high level of this protein in, we can say something about the chances that that bee will be disease resistant and use that information to make a decision on which bees should be bred with which other bees to try and select for higher and higher levels of disease resistance. So we did, and this now takes us back to about 2011, where we started a year project doing exactly as I said. We are going to use these protein molecules, these markers, to try to guide a selective breeding program. But we're going to do that in parallel with a conventional breeding program that uses this hygienic behavior test. And here there's one of the technicians showing you some of the areas that had been killed by liquid nitrogen and how they've been cleaned out. So again, now we're into 2012 and 2013 where most of this work took place. And we went through 3 generations. So we started off with a general population, used the information about how hygienic those bees were or how high the levels of these proteins were in those bees, and crossed certain bees with certain other bees to generate the first generation, went through that process 3 times to generate the third generation, and then all along we were doing many different evaluations on the progeny from those generations to ask whether they are showing any increased levels of disease resistance. We even took some of those bees, gave them to commercial beekeeping operations in BC and Alberta, Saskatchewan and Manitoba, and in a blind test asked them to also test whether those bees needed more, or whether they needed to do additional or less management with those bees versus their conventional bees. And again, it was blind, so they didn't know which ones were selected and which ones were not. There's oodles of data from that project, and I'm just going to highlight a couple of the few findings from the disease challenge experiment that's listed here. So that is, what we did was give these bees levels of disease that would normally knock a colony flat in a couple weeks or a couple months. So there's a disease called American foul brood that I mentioned. It's quite a deadly bacterial disease of bees. Luckily, it's largely under control across North America, but it still occurs in about 1% to 3% of colonies in any given year. And so what we did was take some of the spores from this bacteria, and that's what is shown here. We took an area of comb that had a lot of spores in it, and then made up these frames, put these frames with very high levels of disease into experimental colonies, and asked how those colonies did. Some of those colonies had queens that had been selected through our marker-assisted selection process. Some had been selected through the conventional test with liquid nitrogen, and others had been not selected at all. So I will remind you of what these different terms are. So FAST stands for field-assisted selection. These are the ones selected using the conventional liquid nitrogen test. MASS are the bees selected using our markers and the protein markers. Benchmark are bees that were not selected but were taken randomly from the general Canadian population. And import bees are bees that are from one of the sources of queens that Canadians often import several 100,000 colonies from each year. The levels of mortality are what I want you to look at. So we measured this in both the first generation and the third generation. The bees selected using the conventional test, only 21% of them died in the first generation. That went up, actually, which was unexpected in the third generation. But what we were most interested in was this column where slightly more than half of the colonies died in the from the first generation. But by the third generation, down to 39% of the colonies were dying. And given the level of disease these colonies get, that is really spectacular. This disease would normally kill any colony within a couple months. And that's what was seen in the benchmark. So somewhere between 70 and 75% of colonies would die within a couple months. And within the imported bees, which are probably not exposed to these diseases in their home country as frequently, we see higher levels of death. But that's American foul brood. Oh, so this is another picture of the same stats. That's American foul brood. American foul brood, most beekeepers will tell you, is not that big of a problem. It is, if you've got it, it's a huge problem, but most of the time you don't have it. So it's not something beekeepers worry about a lot. Varroa mites are the things that beekeepers worry about the most. So we set up a similar experiment where we took bees, actually weighed out bees and put equal amounts of bees in different colonies. These mites, or these bees were infected with mites at a level of about 1 mite per every 10 bees. So that's about a 10%, up to even a 20% infection level. And so these are extremely high levels. And if your bees go into winter with those levels of mites, they are nearly guaranteed not to survive. So this is the data. So again, we're only looking at the third generation here. And in the bees selected using the conventional test, they're basically not any different from the bees selected using our markers. But about 50% of the bees survived. And in the benchmark, it was about 15%. And the imported bees that are exposed to these mites in their home country, they're at about 30% infection, 30% of the bees survived. So the really exciting take home from this for us was that this approach that really there were a lot of skeptics, including ourselves at the beginning, whether we could actually get it to work. But we've gotten it to work at the same level, or at least at the same level as the conventional selective breeding process. And this process is done at universities and at certain large beekeeping operations, but generally is so involved that it can't be used by most beekeepers. So what we think we have is a test that should allow bees to be selected for natural variation in a particular trait here. And now we're going on to try to develop tests that will allow us to select for bees, for other traits in bees, including honey production, including aggression or lack of aggression, and including some other disease-resistant traits that we know exist in bees. Okay, so I'm going to jump to areas that are not my research, but I'm doing this to provoke a little bit of discussion. So we'll see where this goes. So invariably, when I tell someone that I study bees, the first question is, what are bees dying from? Or have we figured out what bees are dying from? And this story came out 5 years ago, but it actually reemerged last year, much to my surprise. But there was a report that cell phones were causing bee deaths because someone observed that the die-offs in bees were coinciding with increased use of cell phones. So someone published this study, and what they did was take 2 bee colonies, and they put an active cell phone under one, and they took the battery out of another cell phone and put it under another colony. And then they observed later on that there were more dead bees in the one with the active cell phone under it than in the colony without the battery-operated cell phone in it. And from this concluded that cell phones were causing bee die-offs. So this is literally the evidence behind the claims that that cell phones are causing bee die-offs, and it's hard to explain just how many problems there were with this study. But the very least, there were only 2 colonies. It was not replicated, and it wasn't even clear that they were that the colonies were treated the same way from the study. So there is no evidence that, well, in this case, cell phones, but more generally that electromagnetic radiation of any sort causes bee die-offs. There have been other studies on other sources of electromagnetic radiation, and there's been no confident link established. So there's also been, some of the die-offs have been attributed to other weird diseases of bees. And there was a big story made, I think, 2.5 years ago or so, about something called the media caught onto it and called it Zombees. These, the authors were a little bit at fault here, but the actual science that was done was actually done pretty well. It was just that the media took an interesting story and really ran way too far with it. But what was observed was that you get these little parasitic flies that seem to be resting on bees, and someone found some dead bees by a lamppost and observed that there were these flies on them, and they looked into them a little bit more, and you can actually see a larva of a fly emerging from the bee here. And there's no doubt that these bees, or that these flies, are feeding on bees. The whole zombies phenomenon came, though, because someone observed from the same colony that these bees were very disoriented. They were out at night, and they were sort of like stumbling around. And so the authors claimed this term zombies, and then media had a lot of fun with that. But so that's more to make the point that there are other causes of bee die-offs, and for sure we don't know everything. But you can say for certain that most of the die-offs that we know that we've observed are due to causes that we understand. And this was another cause that we didn't previously understand, but now understand a little bit better, but those flies, or those parasitic flies, are probably responsible for less than 0.1% or 0.01% even of bee die-offs. Now, this is sort of the big topic of the day, neonicotinoids. This is a class of what's called systemic peptides. That systemic meaning they're taken up by the plant through the root system or when the plant germinates and remain stable within the plant for some length of time. Then a pest comes along and eats the plant and ingests the pesticide, and the pest dies. And this is different than foliar types of pesticides where you spray it on the outside of the plant and the pest still comes along and eats it and dies. But because it's systemic and so long-lived in the plant, you need much less of it, and it's quite lethal to the pest that is targeted. These are extremely widely used. There's a couple dozen, I think, different forms of neonicotinoids that are used now in all areas of agriculture, essentially, as well as even houseplants and other plants. And one of the things that makes them so attractive is that they're 10 to 20 times more potent than older classes of pesticides. And they're also, an additional thing is that they have very, very low toxicity to humans. So from a human safety point of view, they're great, really. So what's all the fuss about? Well, there's a lot of things that have linked neonicotinoids to bees. And the word of the day is sublethal effects. And I'll say a little bit more about those later. But essentially, people have observed that, so neonicotinoids are an insecticide. An insecticide, by definition, kills insects. And so if you give a bee enough of an insecticide, including these, it will kill the bee. But one of the things about neonicotinoids is they're also about 10 times less lethal to bees as they are to the target insects that they're used to combat. But other people, or scientists, have observed what are called sublethal effects. These are effects where you give the bee a dose of the insecticide that doesn't kill it, but it causes other problems. The big challenge in the field right now is that nobody really agrees on the best methods to use. What constitutes a sublethal effect versus a lethal effect. And the results are, because of this, the results are really all over the place. Some people show a sublethal effect, others show no effect. And there's always, as always, there's a publication bias, so you only really get to publish things where you see a positive effect. So there's actually a lot of data out there that shows no effect, but it never gets published, so you don't know about it. Now, that being said, I'm not actually a defender of neonicotinoids. I just want to try to paint all sides of the picture. That being said, there are absolute, no question, cases where neonicotinoids have resulted in large-scale bee die-offs. So the first observations were in Germany, but this also happened in Ontario as well, with something called corn dusting. So when corn seed is coated in these neonicotinoids and planted, the hopper that held the seed at the end of the seeding has a lot of this neonicotinoid dust in it, and that gets kicked up, and sometimes the farmer just washes it out into the field. And so there's enormously high concentrations of neonicotinoids where this happens. And bees, there's a few cases where even up to 10,000 colonies of bees have been exposed to this very high level and died. What this means, though, is that the way that farmers are applying the neonicotinoids, and more even how they're cleaning up after they use the neonicotinoids, really needs to change. And there's an effort to try and change the directions that farmers get from the seed companies and from the chemical companies to use these. And that, I think, is eminently addressable. It's these sublethal effects that really we're not very sure about. No question, though, there's also overuse of neonicotinoids. They're often used, or Seed is often just sold with it, regardless of whether there's actually evidence that you need it in that particular area to control some given pest. So again, we have to get a bit better at not just prophylactically using insecticides. But this is I'm going to show you two graphs here that I think are very interesting. So this is the neonicotinoid use in the whole of the United States, and I couldn't find the data for Canada, but I think it parallels Canada very closely. They were introduced in 94, and then there was a big spike as new ones came on the market in 2003, 2004. And we're now up to about 3.5 million pounds used in the U.S. Seems like a whole lot. But these are the numbers for some of the non-neonicotinoid, the classical types of pesticides. And here the numbers are much, much greater. So this is one particular class of chemical where in 95 there were about 15 million pounds of it used just in California alone. And now, because neonicotinoids are more effective, we can use less of them and really are cheaper in many ways. The use of these pesticides has decreased. And the issue with these pesticides is that there's no such thing as a sublethal effect in bees. It's just lethal. So another interesting thing is if you look at the application rate. So chlorothianidin, which is one of the neonicotinoids used on canola, you use it at about, where is it here, 250 to 600 mils per 100 kilograms of seed. And so that means for a very high seeding rate at about 5.6 kilos per hectare. When you use it on corn, you're at about 992,000 milligrams per hectare. 92.5 grams per hectare. You do that once because it's a systemic pesticide and it lasts forever, basically, the life of the corn plant. Whereas chlorpyrifos, when it's applied on corn, you're using it at over a kilogram per hectare, and you have to do that 3 to 4 times per year. So the amount that you have to use of these conventional insecticides is vastly greater than the neonicotinoids. So it's not to say at all that neonicotinoids are good, but they're, I think, a whole lot better than the alternative. So the EU, coming up on 2 years ago, put a moratorium on the use of neonicotinoids. And they did that in response to a lot of public pressure, but they did it without consulting the people that actually need the neonicotinoids, the farmers who need them to control pests on their crops. And there's quite a movement to ban them here, and it'll be interesting to see how that develops. Just today, actually, I got a notification on one of the listservs that there's now a bill before Congress in the U.S. to ban neonicotinoids there. It sounds like it doesn't have much of a hope. But what I've tried to get across to the public, I guess, is that it may sound like a good idea, but if we actually get what we wish for, we'll be very likely to be worse off afterwards, because the lack of farmers' ability to use neonicotinoids is going to mean that they're going to have to go back and use the other chemicals. Now, you can make an argument about changing farming practices, and that, I think, is a whole separate argument. But for the moment, I think neonicotinoids are going to be here to stay. The hope is, or where there is hope, is that there are several other classes of insecticides on the horizon that might be on the market within a couple of years. And because of what we've learned with neonicotinoids and sublethal effects in bees, it's not just bees, but there's other beneficial insects that are affected by these as well. But because of what we've learned from all this, there'll be more scrutiny given to these other classes of insecticides before they get to the market. So hopefully we won't find out 10 years down the road that there's problems with these that we didn't know about. Okay, I've gone on long enough, so just some quick thanks. First to Genome BC for hosting me to come here, but also Genome BC and Genome Canada particularly for funding this research. It couldn't have been done without a team of about 100 different people. But most importantly was Liz Huxter, who is a bee breeder from the West Kootenays in Grand Forks, and Heather Higo, who is a bee breeder in the Lower Mainland, and Steve Purnell, who's an Agriculture Canada scientist in Alberta, and Shelley Hoover, who works for the Government of Alberta in apiculture. I also need to thank my lab, who are the ones who do all the work. I just get to go to fun places and talk about it. And so I will end there and take any questions, but I always end my talks with what inspires me. And it's my kids that inspire me, and being able to go to great places like this. So this is Mount Robson, a little ways north of you here. So thanks very much for listening, and I'll be happy to answer any questions. Yep.
What kinds of bees are safe to touch?
So if I don't know if the question was recorded, but the question is what kind of bees are safe to touch. So honeybees, there's male and female honeybees actually, and only the females sting. So if you can look at a honeybee and find a male, it's safe to touch. I encourage you to be very familiar with what a worker bee looks like versus a drone before you try that, though. Wasps and hornets obviously will sting you, but most of the rest of the bees are not stinging. But often, most of the time, you wouldn't recognize them as bees either. They look quite a bit different than what the average person thinks of as a bee. Yeah, that's a really interesting question. So, should I be repeating questions? Would that get recorded? Repeat it? Yeah, okay. So the question was, are there differences in the hygienic behavior between the different lines of bees or the different subspecies of bees? There are somewhere around 24 or more subspecies of bees. Canada, we sometimes think of us as having certain subspecies, but really Canada and the US have been largely isolated, so that there's no longer really distinct subspecies within Canada or the US. But that being said, I can still answer the question. So the short answer is no. The longer answer is that it's a little bit biased because some of the selection that has been done for hygienic behavior that has actually made it into the North American stock was done on certain subspecies. And so I think it's more an effect of history there, that selection was done, and then some of that genetic material propagated through the continent, rather than the original European sources of those bees being more hygienic than others. Yep. Yep. Yeah, so that's a whole other area of research in my lab that I think is really exciting. I should add that I glossed over a few facts there, one of which was that there was actually 13 different proteins, and I just showed you two, just to give you a sample of what the data looks like. But there were 13 different proteins that correlated with the hygienic behavior. We know what genes they correspond to, and we know something about the function of most of those proteins. And what is really interesting is that at least 3 of those proteins are involved in odor detection. And it's thought that the main mechanism of hygienic behavior is the bees being able to detect an odor that's either coming off of the disease itself, the pathogen itself, or that the dead or dying bee carcass is emitting. And so that's the basis of a hypothesis, really. We haven't proven any of that, but the hypothesis is that these odor-sensing proteins are probably involved in detecting some volatile compound that's coming off of either the pathogen or the dead larva. There's also actually those two proteins that I showed you, those ones in particular are involved in nerve transmission as well. So again, for any behavior, obviously nerves are going to have to be involved somehow, and we expect that the antenna of these bees or we hypothesize that the antenna of these bees, these hygienic bees, are more sensitive to whatever compounds are being released from the dead larva, and then either they're more sensitive in being able to detect that compound, or they're better able to transmit the signal from the receptors on the antenna to the brain to stimulate a behavior. So there's the proteins, I think, that we found are completely non-random selection of proteins from the bee genome, but there's a group now in my lab that's trying to follow up and really figure out what role those proteins play in the function of the hygienic behavior. Here?
I'm wondering if when you're doing the selection process, have you noticed any changes in the bee exoskeletons or?
Yeah, so we haven't gotten to that level of detail. One concern, well, I mean, it was an obvious concern, but it was something we tried to pay attention to, is that the problem with selecting for one trait is that you may select against other traits, and that may be what, or are you getting at it that we may be selecting for bees that basically have harder armor and are better able to resist disease that way?
I was wondering if there's going to be some maladaptions to the selection.
Right. Okay. Sorry, I also realized I didn't repeat the question. So the question was, do we see any differences in the exoskeleton, perhaps because we're selecting against, or the bees that we're selecting for become maladapted to in some other way? And this is a big concern in artificial selection. If you select too strongly for something, you may select against something else. So we didn't look at that question in particular, the exoskeleton. That is something that could be measured. We've looked at other things, though, like honey production and aggression in bees, which are two other things that beekeepers are very worried about. And basically, we don't see any differences. Now, the caveat there is that 3 rounds of selection is not very much. So if one selected more, then perhaps you would see some detrimental effects. And it's known that in groups that have tried to really select extremely for hygienic behavior, they do see higher levels of aggression. So the big lesson there is that you cannot select for one thing in exclusion of all the other considerations. You have to be able to use these, or use any kind of selection. It has to be integrated into a broader breeding program where you're still selecting for other things that are useful. But what we're hoping to get to is a state where we can basically select for all of the traits that beekeepers find to be useful, and that will still have to be built into a wider selection program where you're bringing in outside genetics to keep the gene pool diverse enough. There's another question over here.
Yeah, I mean, you've almost answered it, because I was going to ask how you see the future of your research for the beekeepers or
Yep.
the bees. So you said it already, you hope that you can accomplish all this. How long do you think that's going to take? I mean, when will the beekeepers possibly get the perfect resistance?
I try not to use the word perfect. So the question was, first, how long is it going to be before the average beekeeper will have access to this kind of, to be selected in this way? So actually, we're starting 2 things this summer. One thing we're hoping to start is this bigger project to look for other markers of other traits. But we're also taking the data that we've got, because we've already been working with some bee breeders. This summer we're going to be starting to work with some bee breeders around British Columbia to start using these traits to incorporate, or these markers to incorporate selection for hygienic behavior into their bees. It's, I guess, where one has to start to answer your question is to take a hard look at actually how far you need to go, because you shouldn't need a bee that is absolutely 100% hygienic in order to resist disease. And in fact, We already saw with the varroa mite challenge experiment that we did that we're getting to a level where probably within the third generation, we've got bees that would be able to resist basically any normal level of varroa. The levels of varroa mites that we gave them were completely unnatural. So we gave them extremely high levels, and they were still able to deal with that. So the next question to ask, I guess, would be in a broader study, can you get away with that third generation without treating for varroa at all? And my guess is that you should be able to, because the levels that they were able to deal with already would suggest that if you've got 10 times lower levels, which would be normal, then they should do quite fine. And you should bring your losses down to somewhere around 10 to 15%. We don't really have any hope that we'll get better than 10 or 15%, because beekeepers there's a lot of knowledge built in the beekeeping community, and the average Canadian beekeeper ever gets is about 10%. So you're just always going to have to accept some losses. The industry is set up to deal with that. So if we can bring it down from 20 to 30% down to 10%, then that's what the industry needs. But we haven't done the tests about where we're at now would be good for those.
That's it. So again, thank you very much for joining us tonight. Thanks everybody for coming. And we do have a for you. So thank you from the college.
Thank you.
And we look forward to seeing you next year.
I just want to say thanks again too, but if anybody has any questions or wants to ask about anything offline, then my email, Google me at UBC, and that's the easiest way to find me. But I'd be happy to talk with anybody who's got any questions. Thank you.